Thanks very much, Saskia. Thank you all for having me here. Very excited to be here. It's an honor and a pleasure, and been really impressed by the work that I've seen so far. Today I want to tell you a bit about my research studying vision, choice, action, and engagement. I'm interested in understanding how these processes work across the brain, what the neural mechanisms are. You know, what is happening in your brain when you feel thirsty and reach out to pick up your cup of water and take a drink? What is happening when someone raises their hand in the audience and you orient to them and start to listen in on their question? What parts of the brain, what circuits, what mechanisms underlie these behaviors? And if you go to a lot of neuroscience talks, you probably have a pretty strong expectation about the part of the intro of this talk that comes next, which is that I should pick a particular brain region and tell you a little bit about how cool that brain region is and what the experiments that have been done on that brain region are in the past, to sort of link it into some of the aspects of the behavior that I just told you about. I think, you know, it's clear that we have learned really a great deal in the history of neuroscience by this kind of serial study of individual brain regions and their roles and behaviors. But I think if we take a step back and take a more holistic view, we can recognize that this approach has some limitations, because we've known for more than 100 years that all of the different brain regions, in this case, you know, looking at a sagittal section of a mouse brain, all of these different brain regions are bound up in anatomically recurrent loops that interconnect them deeply and in recurrent ways, and that even when a subject is at rest, these structures produce activity that has complex and unpredictable dynamics. And so I think, you know, I would submit to you that in view of such facts about the nature of the system, that we should not have any prior expectation that it should be possible even to understand fully how a behavior works or what the role of any one of these brain regions is in isolation. So, you know, in the way that you wouldn't try to understand a combustion engine by studying spark plugs without considering fuel, you wouldn't try to understand harmony by listening only to the violins, I don't see why, in principle, it should be necessarily possible to understand the activity or the functions of any one of these brain regions, considering them in isolation from the other brain regions that they're embedded in, the other circuits, the broader circuits that they're embedded in. Today I want to tell you about experiments that we've done, taking advantage of novel tools to be able to sort of take this broader view and look across brain structures in the context of a single behavioral task and try to understand the context in which different brain regions have their functions. You may think, you know, well, how about you just take a part of a behavior, just study the vision, just study the action execution, then surely we can focus in on one brain region and understand the role that brain region plays. But I think what you'll see in the data that I'll show you today, and really what we have known also from the literature, is that it's not the case that the functional signatures or mechanisms that underlie even parts of behaviors are neatly localized to individual brain regions. And so that's the sort of big picture story that I want to convey today. Okay, so to start with, I'll just describe the tool briefly that you all know about, which is the Neuro probe. That has enabled us to really look with a broader view at without sacrificing resolution across brain circuits. And so the Neuro probe, a project in collaboration, of course, with the Allen Institute here, is a CMOS recording device. So the magic is that the computer chip with all the amplification, digitization, and multiplexing hardware is right there on the probe, which enables you to record densely from hundreds of recording sites with a probe that has a very small device size. And for the aficionados, here showing raw voltage traces from a few milliseconds, showing that we can record these high-quality individual action potentials. That is, we have the resolution of single neurons and single spikes. But we have that resolution across major parts of the brain. So we've been able to take advantage of the small device size to record from, in this case, 8 neuropixels probes simultaneously. That's over 3,000 channels in an awake mouse. Very similar, of course, here to the data sets that Josh and others have collected here at the Allen Institute and released recently in a very impressive and exciting pipeline project. And with these data, colleagues of mine, Carson Stringer and Marius Pachitariu, did some beautiful analyses characterizing how the neural activity relates to spontaneous behaviors of the mouse. So our sort of hypothesis going in might have been that we see activity only in motor-related areas of the brain, things that have motor in the name of them, for instance, like motor cortex. But in fact, if we take the neural activity across all of these different brain regions, here showing activity from about 2,500 neurons on a sort of minutes time scale, and try to predict that activity from the observable spontaneous behaviors of the mouse, you can see that actually we can do a pretty good job predicting the activity, the large features of the activity across all of these neurons. And that's true for neurons in many different brain regions, even back to visual cortex here in the cyan. This activity is not just about arousal or overall low-dimensional state, because you can see as we increase the dimensionality of the spontaneous behavioral information, we get more and more predictive power to relate the activity of the neurons to the spontaneous behaviors. And so what we find then is spontaneous behaviors are encoded globally across the brain, across at least all of these structures that we measured from, including multiple different brain systems. But what about goal-directed behaviors? What about behaviors that have a purpose or a function that are trying to achieve some reward? So to study this, we trained mice in a behavioral task in which they see visual stimuli. Here, a simulation of the screen that the mice see in front of them. They see visual stimuli come up on one side of the screen, both sides of the screen, or neither, and they need to choose the higher contrast stimulus. So it's a visual contrast discrimination task, and they can make their choice by turning a little rubber Lego wheel that you can barely see, sorry for the contrast, there. And while mice perform this task, we record with NeuroPixels probes simultaneously across hundreds of neurons in multiple different brain structures. Here I'm showing recordings down the bank of frontal cortex through areas like secondary motor cortex, cingulate cortex, prelimbic, also through primary visual cortex, and parts of the hippocampus and thalamus. And you can see at a glance that there's a great deal of coordinated activity both within and across structures in the context of this behavioral task. So now I'm going to show you some example neurons that sort of illustrate and summarize the sort of patterns that we observed across brain regions. So what brain regions did we record from in total? We recorded about 30,000 neurons in 42 different brain regions illustrated here. And you can see this includes parts of visual cortex, but also frontal and motor cortex. It includes subcortical areas like the striatum and other parts of the basal ganglia, thalamic areas, hippocampus, and parts of the midbrain. So some of the neurons that we observed were like this one here. So this is an example neuron from visual extrastriate visual area, PM. And this neuron, I'm showing in this plot spike rasters from different subsets of trials in the different colors. And you can see this neuron has reliable responses on a few trial types. And what links these three trial types for which this neuron has responses is that a contralateral visual stimulus was shown. So a contralateral visual stimulus is shown in each of these cases, and what differs is the context and the action that the mouse takes. So in this first case, these are contralateral stimulus trials, but the mouse makes a correct choice, correctly detects and turns the wheel to identify that contralateral stimulus. Here, the same stimulus is shown, matched for contrast, but the trial outcome is a miss. The mouse fails to detect the stimulus and does not turn the wheel. And you can see this neuron still responds. And here we present the same stimulus, but now outside of the context of the task altogether. The mouse has no opportunity to even earn a reward if he were to respond, and the mouse does not respond. And this neuron still responds to the stimulus. So this neuron has what we would call a visual encoding. It's not dependent on the particular action that the mouse takes on the trial. It's just dependent on the visual stimulus, and it's locked to the visual stimulus onset. You can see this neuron also has some other activity locked to the movement onset indicated by these black dots, but the initial activity is a visual stimulus locked activity. And we see this kind of visual encoding across areas of what essentially make a classic visual pathway. This includes primary and secondary visual areas, superficial layers of the colliculus, areas like the striatum and visual thalamic nuclei. It also includes some areas like frontal cortex and parts of the midbrain that are maybe a little bit more unexpected to see in the visual pathway, and I'll come back to these areas in a minute. A second type of neuron that we observed are neurons that respond not dependent on which stimulus is shown, but dependent on which action, but dependent on the execution of an action. So this neuron, you can see, responds around the time that the movement starts. And if I align to movement down here, you can see that this activity is aligned to movement onset. But it doesn't matter which action the mouse takes. This neuron is responding to the fact that the mouse has turned the wheel, but whether it's a contralateral and ipsilateral, that is a left or a right wheel turn, doesn't matter to this neuron. And this kind of non-selective action encoding is global. And I think this is a really important finding here, consistent with what we showed about the spontaneous behaviors being encoded across the brain. We see that action encoding, non-selectively at least, is really globally found. And I think this is something that's important for all of us to realize when designing our experiments and when interpreting data from neuroscience experiments. So that you see this non-selective action encoding everywhere. Okay, but we did find some selective action encoding, that is choice encoding. So this is a neuron recorded in the zona incerta, an area of the midbrain, that responds locked to the time of movement onset and around the time of movement onset and strongly and reliably, but only for one of the two choices. So this neuron cares about whether you make a contralateral or versus an ipsilateral choice. And we found these neurons much more rarely, but in a few key areas. And so here I'm showing you 8 example neurons, 4 from the midbrain, 2 from the midbrain reticular nucleus, 2 from the deep layers of the superior colliculus, and 4 from the forebrain, 2 from the secondary motor cortex, this is sort of frontal cortex of the mouse, and 2 from the CP, that's caudiputamen, the striatum, the input structure of the basal ganglia. And you can notice 2 things about these responses and the way they differ between the midbrain and the forebrain. First is that all of these midbrain example neurons I'm showing you, and this was borne out across the population, prefer the contralateral choice, whereas in the forebrain, it's about 50-50 whether they prefer the contralateral or the ipsilateral choice. And second, the midbrain neurons are all suppressed below their baseline activity for, not all, sorry, many of them are suppressed below their baseline activity for their non-preferred choice, for the ipsilateral choice. Whereas in the forebrain, neurons are primarily enhanced for both choices, just more for their preferred than the non-preferred. And so we characterize this as a bilateral encoding in the forebrain, in the frontal cortex, and in the basal ganglia, versus a unilateral encoding in the midbrain, including these unexpected structures like the midbrain reticular nucleus and the zona incerta here and here. Suggesting, for instance, that there may be a more cooperative process between the left and right hemispheres in the forebrain, and a more competitive process going on between the left and right hemispheres in the midbrain. And finally, I mentioned that we presented the stimuli outside of the context of the task, and when we compare the activity between these actively engaged responses when the mouse is doing the task and is trying to detect the stimuli and respond to them to earn rewards versus the passive presentations, we noticed a striking difference. We noticed that while the visually evoked responses were not radically different between the two conditions, we noticed that actually there was a difference in the baseline offset, so the shift, a tonic shift in the average activity across neurons that was consistent across systems, specifically a suppression of activity in cortex versus an enhancement of activity in subcortical areas, excluding notably visual thalamic areas, which instead had the cortical pattern of being suppressed during engagement. And so we think that this might be reflecting a need for enhancing the signal-to-noise ratio by suppressing baseline background activity in cortex, but bringing subcortical areas closer to the threshold they need to actually trigger a decision and trigger a movement. And so we think that this global organization of the engagement correlates may reflect an organization of the function of these different systems. So in sum, by looking across these 42 different brain regions, across tens of thousands of neurons and characterizing their correlates, we have identified these sort of organizing principles of the behavioral correlates in the context of this task. We've identified that action encoding is global, seen everywhere that we looked. We identified that choice encoding is much rarer, but is still distributed across multiple systems in cortex, basal ganglia, and midbrain, but with a difference in encoding between a bilateral encoding in the forebrain and a unilateral encoding in the midbrain. And finally, we observed that engagement differentially influences or differentially is correlated with cortical versus subcortical areas with an enhancement of cortical areas plus visual thalamus and a suppression of subcortical areas. All right, so in the last, I'm going to now touch on 2 other topics, that one of which is a future direction where we're trying to develop new versions of the NeuroPixels probes in collaboration with researchers here at the Allen Institute. And the other is we were asked to talk about our big team in open science, and as Saskia mentioned, I'm part of the International Brain Lab, so I'll give us a couple slides on that project as well. So first, thinking about future of NeuroPixels. So there are neuropixels, I think, in my opinion, are amazing, and I think we can really collect transformative data as I've shown you here, and as people at the Allen Institute have done, and we saw in some beautiful posters, for instance, from Sam yesterday. There are some remaining data quality issues that I think we need to work on if we want to improve the technology even further. So first of all, the brain is not static. It's floating in CSF in the skull, and it moves around. And so here I'm plotting a spike raster. X-axis is time. Y-axis is actually the position of each spike as it occurs along the length of the probe. And you can see this coordinated motion of all of these spiking neurons up and down the probe. This is the brain literally moving up and down the probe. And so there's this instability, which is a problem for spike sorting. Second, with electrophysiology, of course, we have minimal cell type information. Classically, people have discriminated essentially a biophysical discrimination between narrow and broad spiking waveforms. That gives you just the first bit of information about cell types, bit taken literally. And finally, we recognize that there are sampling biases in electrophysiology, that some small neurons like this cerebellar granule cell here that have exceptionally small electrical fields may be difficult to detect with normal electrophysiological technologies. Okay, so I'm going to tell you now about a new version of the NeuroPixels device that we're developing that is attempting to overcome these limitations and produce improved measurements of brain activity. So this device is not a superhero movie, but it is called NeuroPixels Ultra. And the Ultra here refers to the ultra-dense recording site sizes. So on the left, I'm showing the recording site size and spacing in NeuroPixels 1.0. These are 12 by 12 microns with these sort of hefty gaps in between them. Whereas on the NeuroPixels Ultra, the device design that we have developed is 5 by 5 micron sites with very small 1 micron gaps in between the sites, thus covering a sort of two-dimensional plane of recording site area. And for the aficionados who might wonder about what happens to the impedance, and that's the noise levels in this situation, we can observe that because of the impedance characteristics of the recording site material, titanium nitride, and because of the way that the noise levels are primarily dominated by ADC noise and not electrode noise in the 1.0 devices, we can actually go quite small with the recording site sizes down to this much smaller site size and still have good noise characteristics in NeuroPixels Ultra. And yeah, let me just say, so this is a project that's now funded by the Brain Initiative, and it's in collaboration with Sean, Josh, Xiaoxuan, and of course Christoph here, and also with our collaborator Tim Harris at Janelia. Okay, so here's some example data from a pilot device about what we can detect with this kind of recording device. So what you're seeing here is real mean waveforms recorded with a pilot device from a few example neurons, and these are waveforms averaged across the multiple spikes of the neuron, and each black line is the waveform recorded at one of these recording sites. And so you can see a few things immediately. One is that we can detect some really small electrical fields, like this neuron in the striatum that has apparently an electrical field of only about 5 to 10 microns diameter. You can imagine, now that we're getting these really electrical images of the electrical field, that we can actually borrow techniques from computer vision and from two-photon imaging, where we can do stabilization of the recording post hoc computationally via image registration. So if the neuron moves up and down relative to the probe, we can literally still see it and still track it. There are no gaps for it to fall in, and we can computationally process that afterwards. And finally, you can see that in these examples, we're getting not just information about the biophysics in terms of the shape of the waveform shape differs, of course, between here and here, but we also can see morphological information. You can actually see some of the subcellular processes of these neurons, and we think we can use that to give us an extra bit or two of information about cell type, which I think will allow us to identify more cell types than we are currently. Okay. So last couple of slides, switching directions again. So I want to just say a few words about the International Brain Laboratory, which I think is a really exciting large-scale collaboration, in many ways modeled after aspects of the Allen Institute and trying to provide a slightly different model to tackle some of the same big picture questions that the Allen Institute has gone after as well. Okay, so what is the International Brain Laboratory? It's a consortium of of about 20 different laboratories, half experimentalists and half theorists. I'll show you their names in a minute, who have a shared common goal, which is to study a single behavioral task, a single behavior, in a standardized way across labs, collecting data in a standardized way, sharing that data immediately amongst the labs, and analyzing it in a shared, coordinated fashion in order to come to sort of a broad consensus across many different viewpoints about the nature of decision-making, the nature of this behavioral task across these labs. So this involves standardizing the experimental apparatus. This is a 3D rendering of the standardized IBL behavioral apparatus. By the way, you can go to the website and download the plans for this and build one yourself if you like. It involves standardizing, settling on a shared behavioral task that is standardized across these labs. In this task, very much like the task I showed you, mice see a stimulus either on the left or the right and turn the wheel to indicate where they saw the stimulus. But in this task, there's a key manipulation that is changing the probability that the stimulus appears either on the left or the right in blocks. And this invokes a couple of cognitive processes that the IBL is really interested to study. One is integrating past history information to identify where the stimulus is likely to occur, and the other is combining that prior information then of an expectation about where the stimulus occurs with the current sensory information in a kind of Bayesian computation to identify the most likely decision that will lead to reward. And so I think these are really interesting cognitive processes that the IBL task will get at. And after a great deal of standardization, literally down to working out differences in the diets of the mice across institutions, et cetera, the IBL has collected over 1 million behavioral trials here showing psychometric curves from 7 different labs that have now gotten to the point where everything is sufficiently standardized that that the variance within labs is greater than the variance across labs, and so you can see that the distribution of the psychometric curves is very consistent across labs. I mentioned that the key aspect of the IBL is this collaboration between experimentalists and theorists. The data is all immediately standardized and uploaded to web servers. And a description of this data architecture has recently been posted on bioRxiv, so you can read about exactly what that process is like and what the standardization is like. And in fact, this website just launched recently, and so you can go there and see not yet neural data, but you can see behavioral data and download behavioral data from many of our mice and access the output of the IBL directly. And this is where neural data will appear hopefully sometime next year. So what neural data is the IBL going after? So in the first pass, the IBL is attempting to make a brain-wide recording map with neuropixels. And so this is a radically higher density set of recordings than what I showed you earlier in this talk, and obviously in this even more interesting version of the task. And so I'm super excited about this. What you're seeing here is slices of the brain with the planned recording trajectories. Each black line is the span covered by 1 neuropixels probe. And you can see over some hundreds of recording penetrations, which will be shared across these 10 different experimental labs. The IBL plans to cover densely the entire mouse brain, hundreds of brain regions. Okay, so the IBL is a huge collaboration, and I just want to show the size of the team that's involved. These people are all of the postdocs and technicians who are really driving forward the IBL. I want to especially note Olivier Winter, Nicolas Bonacci, Gaël Chapuis, and Cyril Rossant, who are the 4 full-time engineers of the IBL, and who have really put in outstanding work to bring together the data architecture that I showed you and the standardized behavioral setup, et cetera. And these are the labs that are involved. Great. So that's all I have. Let me just conclude. So I showed you that with NeuroPixels, we have this ability to really scale up to look at across brain regions, but without loss of resolution. And I think that's really exciting for the field. And in particular, it's enabled us to identify these principles that organize the behavioral coding across the brain, specifically that spontaneous and goal-directed actions are encoded globally, that choice is encoded in a restricted set of areas and differently in the midbrain and the forebrain, and that engagement correlates with suppressed activity in cortex and enhanced activity in subcortical areas. And finally, I showed you an exciting project in collaboration with the Allen Institute to develop a new generation of device that will enable more detailed, stable, and cell type specific measurements of brain activity. So I want to thank Matteo Carandini and Kenneth Harris, my postdoc advisors, Peter Zakahas, a super talented graduate student who I worked with on much of the work that I showed you here. Tim Harris obviously has led the NeuroPixels consortium, and thanks certainly also to the Allen Institute for Brain Science for their funding of the NeuroPixels project. Thanks.
We have time for a few questions, so if you raise your hand, we'll get microphones to you.
Nick, when will...
Wait for the microphone.
When will the data, the IBL data, be available to the rest of the world, and will that be NWB 2.0?
Yeah, great question. So, so one of the exciting things about the IVL is that it's really an experiment in scientific organization, and there's a really great deal of policies and procedures that have been written, and it's all documented. So the policy on this, the IBL policy, is the data are released 1 year from the date of acquisition or on publication, whichever comes first. And given that the data sets for the brain-wide activity map, some of the first ones have already been collected, you should expect by this time next year to see neural data. Not the whole map maybe, but some of the data set will be released by then.
And it'll be in 2.0?
So that's a good question. I personally have just been working on converting the data sets that I showed you that I collected into NWB format, and we plan on sharing those when that manuscript is released. And I'm just getting up to speed, so maybe I'll ask you more later about the difference between 1.0 and 2.0 on NWB. But I think certainly the intent is that we release in the most modern and up-to-date version of the standards, yeah.
Hey, Nick, so for the first part of your talk, I'm curious about the dynamics of the signal that you were able to record. Did you see some systematic patterns of, it being initiated in one place and then propagating to the other and ending up somewhere else?
Yeah, great question. So, so I didn't get to show those analyses, but we did a couple of different analyses to look at the timing of choice-related information, the timing of activity, especially focusing on this comparison between the bilateral forebrain choice selective areas and the midbrain unilateral choice selective areas. So we did a population decoding analysis, which showed a similar time course of decoding when decoding choice from those two sets of areas. We could not statistically distinguish the time courses. And then we also used an extension of the joint peristimulus time histogram analysis, which is a classic way to look at the relative timing of two individual neurons. We extended it using canonical correlation analysis rather than just spike counting to be able to look at the relative timing of two populations of neurons linked to a particular event, in this case stimulus onset and movement onset. And in that analysis too, we could not distinguish a difference in timing between the frontal cortex and the midbrain areas that I pointed out to you, even though we could reliably distinguish differences in timing between visual cortex and midbrain and between visual cortex and frontal cortex. So if there are differences in timing of the choice signal, they're, I would say, subtle, probably less than a dozen or 2 dozen milliseconds. We were not able to detect them with the current data set and methods. And I think maybe a more parsimonious explanation is that it's not the case that the decision is computed in one area and then propagated to others, but instead that the known anatomical pathways that engage these areas from frontal cortex through basal ganglia to midbrain and back via thalamus, that those pathways form a recurrent loop that instead sort of binds these areas, and collectively across them they give rise to the decision. So I think that's probably the most parsimonious interpretation of the data at this point.
Yeah, here.
Yeah, sure.
That was an awesome talk. Thanks a lot, and I'm super excited for the NeuroPixels Ultra. It's going to be awesome. I'm curious if you could say a bit about how you guys are thinking about how interference between these different encodings may be avoided, right? I mean, presumably you wouldn't, if you're representing visual information, you wouldn't want that to be sort of confused by action encoding. Is it different cells? Is it different neural activity, subspace dimensions? How are you thinking about that?
Yeah, I mean, I think that's one of the, the fact that we don't know these mechanisms that keep these signals separate or integrate them is why we have to do this sort of survey in the first place, is because you just can't predict where different signals are going to show up a priori. And I think that's one of the most exciting directions going forward, is trying to understand what mechanisms enforce the signals that are localized to stay localized, and what mechanisms allow signals to be integrated, essentially the gating and routing and communication across brain regions. Yeah, I've been writing grants about it, but I don't know the answer. Yeah.
Thank you, Nick. So I'm a bit surprised by your engagement results, particularly that we find pyramidal neurons are more active when the mice are engaged in the cortex. So I'm
The other way around. The cortex.
We found that.
Oh, yeah.
And you find the opposite. So you see that cortex is suppressed by the engagement.
Go ahead.
So I'm curious, one, what is your interpretation of that? And second, how do we reconcile that with our results that we see? Yeah.
So
and
actually,
I have a second question I'll ask later. Yeah.
So I was actually really excited to see on Sam's poster yesterday that he was showing with the NeuroPixels data in the change detection task the same result as what I just showed. So on Sam's poster, in the cortical areas, they were less active during the engaged state. Sam, that's correct, yes?
But in imaging,
cortex.
we see
And
the
then,
opposite.
and then in the midbrain, there was a difference, which is that Sam didn't see an enhancement of activity in MRN and SCM. I think it's possible that that's attributable to the difference in the task demands. This is something that occurred to me where in this task, we're asking the mice when they need to make a response to initiate an action, whereas in the change detection task, the mice are asked to, what the mice do at least is stop running when they detect a change so that they can begin licking. In any case, I think the similarities were bigger than the differences on the poster
But
that I saw.
so you'll see, we're going to present, not with imaging, it's the opposite, but you'll see. But maybe it's really the task demands. So in pyramidal neurons, they're more active.
Okay, so if imaging shows a different result in NeuroPixels, then in this case, I would probably be inclined to think that measuring baseline activity is one thing that's particularly tricky to do with calcium imaging, because you have to do corrections for baseline activity in the first place. And so I might be inclined to think that the NeuroPixels would be the way to go for this particular measurement.
And the other question, did you try to get a similar map for arousal and pupil diameter?
So no. What we did show in some analyses is that the engagement signature that we showed could not be accounted for by arousal or by spontaneous behaviors or by previous trial reward history. So we did some controls to show that those things, while those also relate to engagement, those could not predict, those could not account for the ability of this neural signature to predict the engagement of the mouse.
I meant more to get a sense.
Yeah, so I haven't done that analysis,
Okay.
yeah.
Thank you.
Yeah, thanks.
One question which I have is regarding the complexity of the behavior. Do you think that it is possible that the incredibly widespread activities which you're seeing which correlate with a particular event are due to the fact that the dimensionality of the behavior you're using is so much lower than etological variables? And are you trying to think about increasing the complexity of the behavior in tasks like these when you measure thousands of neurons simultaneously?
Yeah, so absolutely. For sure it is the case that the behavioral complexity here is relatively low. This is what I would describe as a two-dimensional task. They have three behavioral options that they can make: turn the wheel left, right, or hold still. They have 2 dimensions of sensory input that they need to use to partition that space and make the correct behavior. So absolutely, we would love to do higher dimensional behaviors. I would love to brainstorm with you about what those can be for mice, because I don't think it's a very simple problem at all to design them. But yeah, that's definitely an important future direction. Yeah. In the back.
So related to that, how deeply do you intend to sample on this? You mentioned you were going to go deep, but do you have a target number of neurons, target number of years until the postdocs are done? Or what, when do you know when it's time to move on to the next task?
For the IBL, you mean? Yeah. So for the IBL, the goal is to record from each of these 273 penetration sites twice, once in each of 2 different labs, which we estimate is some months of data acquisition, and then to fill in gaps or do more dense sampling sort of as scientific questions and demands arise. So that's the goal. So this is definitely won't be the only data set in the history of the IBL. This is the first big data set, and then it will evolve from there. That's the goal. Yeah. One last question here. Yes, Steve.
Have you been recording during the training phase of the choice experiment, or is that feasible?
So in this particular case, we took this strategy of doing recordings where we were doing acute recordings, so we could insert multiple probes into different spots each day over a series of days in the mice to be able to sample as many brain regions as possible per mouse. And so for that approach, it's not possible then to record over training days. But I think what we could do is record from targeted areas with chronically implanted neuropixels, which is something that we showed works really well in the 2017 paper. And that would allow us to record over the course of learning, which I think absolutely is a super important direction.
It seems like there might be some serious hints as to causality and what comes first. And
completely agree. Yeah, I mean, one of the interesting observations that we have here is that there's some of these midbrain areas, like the midbrain reticular nucleus, that appear to have what are really visual responses. They show up as part of the visual pathway by our analyses, and it's unclear whether those exist in an untrained mouse versus they arise over the course of training. I think that would be a particularly interesting thing to follow up on. So yeah, great suggestion.
All right, let's thank Nick again. Excellent.